Hey everyone, and welcome to This Explainer. Today, we're diving deep into the incredible biochemical machinery that transforms sunlight into the exact energy that fuels our entire world. We're going to be focusing on explaining the chapter that covers how photosynthesis actually operates in higher plants. It's a complex topic for sure, but we're going to break down these intricate cellular activities into clear, easy-to-understand concepts. Okay, so let's kick things off with a pretty high-stakes question, just to establish why this microscopic process is literally the reason we're alive right now. What would actually happen if there were no oxygen to breathe? I mean, it's a terrifying thought, right? Green plants are constantly pumping life-giving oxygen into our atmosphere, while simultaneously converting light energy into the chemical energy that forms the base of our entire global food chain. Without this process going on behind the scenes, life as we know it simply stops. So here is our roadmap for the explainer. We'll cover 1. The photosynthetic machinery. 2. Catching the light. 3. The Z-scheme. 4. Powering up with ATP. 5. The C4 pathway. And 6. Factors affecting photosynthesis. Let's get into it.
Section 1. The photosynthetic machinery, basically the factory floor.
Let's start our zoom-in approach by locating exactly where this biological factory operates. It all happens inside the chloroplast, and there is a really brilliant division of labor inside this tiny factory. On one hand, you have the membranous system, that's the grana and stroma lamellae. This is where the light energy is physically trapped and where ATP and NADPH are synthesized. It's the light-dependent side of the whole operation. On the other hand, you have the fluid matrix, which is called the stroma. This is where the enzymatic dark reactions take over, using that newly minted energy to actually synthesize sugars. It is just a perfectly divided workspace.
Moving right along to Section 2, catching the light, the solar panels.
Now that we're inside the chloroplast, we need to understand exactly how the plant captures sunlight. It doesn't just use one tool. It actually uses a very specific array of pigments. So the crucial takeaway here is that while chlorophyll A is the star player, the major pigment responsible for trapping light, it relies heavily on a whole team of accessory pigments. Think of chlorophyll B, xanthophylls, and carotenoids acting like a massive network of cellular solar panels. But they also act as microscopic sunscreen. They physically protect chlorophyll A from photooxidation, so the main engine doesn't just burn out under intense sunlight. At the same time, they're absorbing a much wider spectrum of light wavelengths and funneling all that extra energy directly back to the source. It's incredibly efficient.
Section 3. The Z-scheme. Time for some molecular fireworks. With the light captured, we trigger the photochemical phase, jumping right into an energetic molecular relay race. This is mapped out in what's called the Z-scheme, which tracks the exact path of the energy. Follow the electrons for a second. They get excited by light in photosystem 2, jump way up in energy, travel downhill through an electron transport chain, and finally reach photosystem 1. Here, they get re-energized by even more light and travel downhill once again to finally reduce NADP+ into energy-rich NADPH. And you might be wondering, how does the plant replace those initial electrons? Well, it literally splits water. And that is exactly where the life-saving oxygen we breathe gets released into the atmosphere.
To really make sense of this cellular wiring, we've got to compare the non-cyclic Z-scheme we just talked about with the cyclic flow. In the non-cyclic pathway, both photosystem 2 and I work together in a series, which gives us ATP, NADPH, and oxygen. But sometimes only photosystem I is functional. When that happens, the electrons just circulate back through the transport chain. This is called cyclic photophosphorylation. Think of it as the cell's backup generator. Its only job is to pump out extra ATP to keep the lights on, without making any NADPH or releasing any oxygen.
Section 4. Powering up with ATP, the cellular turbine. Okay, let's move to the next crucial step and see how this continuous electron movement actually builds the primary energy currency of the cell, ATP. This brings us to a vital mechanism called chemiosmosis. You can think of chemiosmosis exactly like a hydroelectric dam. As electrons move through that transport chain we just explored, the plant actively pumps protons into the inner space of the thylakoid, called the lumen. This process creates a massive, pressurized proton gradient across the membrane, literally just waiting to be released.
And this gradient is where the real power is generated. When those trapped protons finally rush back across the membrane into the stroma, they have to pass through a highly specialized transmembrane channel called ATP synthase. The sheer force of these protons moving through physically spins this microscopic cellular turbine, forging ATP from ADP. The breakdown of that gradient provides the exact energy needed to power the plant's sugar-building factories. It's a mechanical marvel at the cellular level.
Section 5. The C4 pathway, maximizing the harvest.
Zooming back out a little bit from the molecular level, let's explore how certain plants use all this stored energy to efficiently build food, especially when they're stuck in really challenging, dry environments. Welcome to the C4, or hatch and slack pathway. This involves a beautiful two-cell relay system between the mesophyll cells and the bundle sheath cells. Plants that have adapted to dry tropical regions use this specialized layout to maximize their carbon fixation while simultaneously minimizing water loss. The plant pulls this off by executing a flawless four-step cycle. First is fixation. Carbon dioxide binds to a waiting molecule called PEP in the mesophyll. Second, transport. This newly formed C4 acid acts like an exclusive VIP shuttle, moving deep into the bundle sheath cell. Third, decarboxylation. The acid is broken down, releasing a highly concentrated burst of carbon dioxide directly into the Calvin cycle. And finally, regeneration. A 3-carbon acid returns to the mesophyll to recreate PEP. Basically, it acts like a specialized, high-powered carbon dioxide delivery pump that guarantees the ultimate sugar-making machinery never ever runs out of raw materials.
All right, our final section, Section 6, Factors Affecting Photosynthesis, the External World.
Let's look outside the plant at the specific environmental variables that ultimately control the yield of this incredible biological factory. You see, a plant's productivity is a delicate balancing act dictated by the environment. First off, light. Light intensity determines the baseline rate, though, as we learned, extreme highs can cause damage. Second, carbon dioxide. This is actually the major limiting factor in nature. Most plants are essentially starved for it, which is exactly why higher concentrations can dramatically boost agricultural productivity. And third, temperature. Temperature strongly affects the enzymatic dark reactions. This perfectly explains why C4 plants thrive at much higher optimum temperatures than standard C3 plants. Understanding these precise limits is absolutely crucial if we want to manage global food yields effectively.
To wrap up our explainer today, I want to leave you with a bit of a provocative thought about the massive global scale of the microscopic processes we've just uncovered. Rubisco, which is the main enzyme responsible for grabbing carbon dioxide in the Calvin cycle, is the single most abundant enzyme in the entire world. But do you ever wonder why? Why does the Earth need so much of this one specific protein? And what does its delicate, constant competition between oxygen and carbon dioxide mean for the future of our green planet? Keep exploring the chapter, and I'll leave you to ponder that vital question.